Right. Good evening, my dear students. So I hope this is the day five, day five of session which I am conducting for advanced level grade 2028 biology students. Right. So up to now we have completed until disaccharides. Right. Disaccharides of unit two. Right. So now last week I introduced you all to the topic carbohydrates. Right. Carbohydrates means they are coming under an organic molecule, the most abundant type of organic molecule. Right. So if you see last week, this is what we discussed. Right, carbohydrates, the main basic features were discussed, and after that we understood there are certain chemical formulas related to carbohydrates, and then we understood that carbohydrates differ, or they have been categorized into three types. What are they? Monosaccharides, disaccharides, and polysaccharides. Right. Mono means one, so they are one, a single type of basic type of carbohydrates. Di means two different type of sugars combined, or two similar type, just two types of sugars combine to form a new sugar. That's the meaning, right? Polysaccharide means, poly means multiple, which means multiple number of sugars combine to form a sugar. That is polysaccharide. And then we came on to monosaccharides. Monosaccharides, there is general formula CH2ON, and this N number varies between 3 to 7. Right? They have certain characteristic features, and then we went on to each type of monosaccharide based on their number, which is N. Right? If the N is 3, they are called triose. There's an example. If it is N equals 4, they are called tetrose. If it is N equals 5, pentose. And then if it is 6, hexose. And then we went on to the structure, how it appears. It is appearing in ring shapes and linear shape. Right? And then we went on to the disaccharide part. Disaccharide part, how it forms, how a disaccharide is formed. Two monosaccharides combine by a condensation reaction to form a disaccharide. During that, what type of bond is formed? Glycosidic bond is formed. See here, this is what is meant by glycosidic bond. See, this is glycosidic bond. So glycosidic bond means, now between, right, if you see here, this is a monosaccharide, this is a monosaccharide, so both the monosaccharides react. Right, here an OH group, here an HOH group will be reacted, and a water molecule will be removed. Here, see, this is the water molecule, and this bond is called glycosidic. Right, the bond that joins two monosaccharides. Right, and then we went on further uh, to understand the reaction that take place. Two monosaccharides combine. So if you see here, glucose reacts with another glucose to form a maltose sugar. Maltose is a disaccharide. And we understood that glucose reacts with sucrose to form a fructose. Fructose is a disaccharide. So these are monosaccharides, these are disaccharides. And we understood a concept called reducing sugar. A sugar which can react with Bredick solution and give a brick red precipitate is called as a reducing sugar. These were the things which we discussed the last week session. Right, I hope these information which I discussed last week was clear to you. Right, I hope it's clear. Right, so today we are going to, you know start a new topic, which is polysaccharide, which means multiple number of sugars combined to form a new type of sugar. Right, okay, let's see. They are polysaccharides, they are macromolecules and biopolymers. Right, they are macromolecules and biopolymers. So what is meant by macromolecules? Macromolecules mean large molecules. Macro means large. Large molecules. Biopolymers means, bio means biological. Polymers. Polymers means, uh, you know, I hope you have come across these, right? Al, do you find any, delay in the writing? Or is it with the flow? Can I just drop a message? It feels like there's a delay in writing. Can you see? Or is it fine for all? All right. Now fine. Hmm. Right. Sorry. Right. Right. You have come across alkene, alkyne. Wait a minute. This is small. Mm. Right. Alkene, alkane, alkyne. Right. Now these are type of, can remember those we understood under uh, polymers, right? They can form polymers, right? So polymers means simply, a collection of molecules, right? The biological polymers, which means biopolymers, right? Polysaccharides are made up of few hundred to few thousand monosaccharide subunits, right? They are made up of few hundred to few thousand monosaccharide sugars, right? These are the characteristic features of polysaccharide. They are non-crystalline. Monosaccharides were crystalline. Now just think, if you take sugar, right? Sugar looks like now normal sugar which we find at our residence, right? At our homes, normally the sugar that we find, it has a crystalline shape. Am I right? It has a crystalline shape. But if you see polysaccharides does not have that crystalline shape, it seems. Right? That is the thing they are saying non-crystalline. Can you get the point? Is it clear? Students, if you have any problem, please let me know. If you can't see the screen, if there is any technical issue or something, right? If you can't hear the voice, is it clear? The feature non-crystalline. Yes or no? Yes. Very okay. Right. Water insoluble, which means normally sugars are soluble in water. They can react with water. But polysaccharides does not react with water. They remain as a suspension. Right? This is the thing. If you see this, right? This is a water. Right? There's a glass of water. And I'm doing is I'm adding a polysaccharide. Right? If I add a monosaccharide, what happens? It will be completely dissolved so that I cannot differentiate the sugar and the water. Whereas a pen, right? But if I add a polysaccharide, what happens is it forms a precipitate. Sorry, sometimes it can form a precipitate, sometimes it will be forming a, suspension. Suspension means like a mist mix, like this. It can form a something like this, right? That is the thing. So it forms like a heterogeneous type of mixture, not an homogeneous, right? Not considered as sugars. They are not considered as sugars, right? So these are the three main characteristic features of polysaccharide, right? Okay. Now, wait, wait a minute. Right. Now this is the thing. You see, I'm taking polysaccharides. Polysaccharides can be separated based on two factors, right? Mhm. Based on two factors. First one, it can be based on the function. Second one, it can be separated or classified based on its form. right? Mhm. Based on its form, right? So depending on the function, it is separated or classified into two as structural, storage. If you see form, it is divided as linear and branched, right? So on structural polysaccharides, we have certain examples. Let's see what are they. First one, cellulose. Is this polysaccharide new to you all? Structure. Cellulose. Where can we find this cellulose? Normally, cellulose are found in plant cell walls, right? Cellulose. Second one, a new one for y'all, which is hemicellulose, hemicellulose, right? Third one, we can say hemicellulose, pectin, and there are so on, right? Let's see. So storage, under storage, we have glycogen, starch, hmm, right? One is glycogen, and the other one is starch. So let's see what are those storage structural parts. Right, we'll be seeing those now. Just for the sake of introduction, I'll give those. Just I'll name them. Form, if you see, polysaccharides can be linear or branched. Right. So linear, if you take cellulose, is considered to be a linear one. Right. Whereas there is another one called amylose. If you see branched, hemicellulose is considered to be a branched one, right? Hemicellulose, amylopectin, right? And glycogen, right? You see, polysaccharides. Polysaccharides can be classified into two based on their function and based on their form, right? If you see the function, polysaccharides are used as a structural component, and second one, they are used as a storage component. For examples for structural components, cellulose, hemicellulose, and pectin. Right? If you find it bit difficult to understand what are these are examples of polysaccharide. Right? We have a separate table to learn about them. Let's learn about them there. Right now, just for the sake of classification, I'll introduce those terms. If you say storage, these are the polysaccharides which are involving in storage components, storage functioning. Glycogen is stored in human body, animals, right? Starch is present in plants. It will be storing. You might ask, so what does it store? I'll explain those. What does it store? How it is contributing in structure? I'll explain those. Now you have to just understand. Polysaccharides are classified into two based on their function, form, and these are the types, right? Okay. Form, if you see the external appearance, how polysaccharides look like, they can be looking like linear or branched. If they are looking at linear, examples for linear, cellulose is linear, amylose is linear. If you see branched, hemicellulose is branched, amylopectin is branched, glycogen is branched. Is that clear? That classification part, is that clear? Students, you can use the chat. If it's clear, you just drop a message, so it's clear. Or not, you just drop a message, so I didn't get it, just explain it again. I'll explain. Is it clear? Mm. Yes or no? Now, for the students who joined bit late, now we are starting about polysaccharides, right? Polysaccharides means uh, type of sugars or polymers. Normally, poly means we didn't get used to those terms, polymers, as polymers, right? Right now, polymers. These polysaccharides are some sort of polymers which are formed by combination of sugars, right? Few hundred to thousand number of sugars like monosaccharides, they'll be combined into form a polysaccharide. So polysaccharides, we are just now getting just we are going through the polysaccharide part. So polysaccharides are based on, they are classified into two based on their function and form. If they are classified based on their function, they can be found as structural and storage, which means simply polysaccharides are used as a structural component and it is used for storage purposes. Examples: cellulose, hemicellulose, pectin, storage, glycogen, starch. If you consider their morphological or external appearance, polysaccharides can be appear as linear or branch. Examples for linear: cellulose, amylose; branch: hemicellulose, amylopectin, glycogen. You might ask, so these are new words. Only we know cellulose, we know glycogen, but others are new to us. We know starch, right? So others are new to us. How they contribute, what are their functions, all of these, right? We'll be learning now separately in a table. Now, what I want is, can you understand this classification? That's it. Is it clear? I explain it again. Is it clear now? Oh, you want me to explain it again? A student has dropped me a message saying, "Sir, can you explain it again?" I just now explained it. If you want, I'll explain it again. Do you want an explanation again? Don't hesitate to ask. I'll explain it again if you want. Hmm. Do you want me to explain it again? Yes or no? You want me to explain or no? Okay, I'll explain it again. No issue. Right? Now this is the thing. Last week we studied about uh it. Last week we studied about a new topic, which is monosaccharide, carbohydrate, right? Normally, carbohydrates are organic molecules, right? Now, I have posted that last session in our YouTube channel, right? I'll share that YouTube channel to you all in the group, right? Now, there what we learned is we learned about monosaccharides and disaccharides. You might ask now, so what is, we didn't watch the video, so what is those? You are saying monosaccharide. Now, this is the thing. Carbohydrates are normally organic molecules, right? These carbohydrates are classified into three: monosaccharide, disaccharide, polysaccharide. So mono means one, mono, right? So that means they are single type of sugars, one type of sugars, right? If you see under monosaccharide, we'll be studying about glucose, galactose, fructose. These are not new to you all. You have learned this in your O levels, right? Now, if you see glucose, if you take glucose, the glucose will be containing only glucose in it. You understand? It won't be containing any other contamination in it, any other sugars in it. The glucose won't contain galactose in it, which means it is pure, right? If you see disaccharide, disaccharides are formed by a combination. Di means two, di means two, which means there's a combination between two types of sugars. Now, example for disaccharide is sucrose. Sucrose is formed by a reaction between glucose and fructose. Glucose and fructose react to form a sucrose sugar, which means two types of sugar. And if you see polysaccharide, polysaccharides are multiple types of sugars combined to form a polymer, right? So here we learned about monosaccharide. Monosaccharide is the simplest type of sugars, and they are classified based on a number, right? If you see, this is the normal. See here, this is a chemical formula for monosaccharides, right? And here the value n, that is the one that is a variable which can vary from sugar, right? So that n value varies between 3 to 7. If the n value is 3, they are called triose. Example, glyceraldehyde. If it is n equals 4, it is tetrose. N equals 5 is pentose, right? If n equals 6 means hexose, right? We learned about few features. They are reducing sugars. Reduced sugar means they are the sugars that can reduce Benedict solution. We learned about these all, right? So those were simple information. If you want further explanation, just go and check the YouTube channel video, right? Okay. And we were learning about these forms, right? They have ring forms, aqueous forms. You don't need to memorize these structures, right? And we went on to disaccharide. Disaccharides are formed between two reactions, between a two there's between a reaction between two monosaccharides, and they'll be forming a disaccharide, right? And then we learned about the glycosidic bond, which is the bond that connects two sugars. And then we came to the final part, which is the polysaccharide part, right? Now, polysaccharides are macromolecules, biopolymers, which means macromolecules mean large molecules. Biopolymers means they are biological polymers. Biological polymers, right? Polysaccharides are made up of few hundred to few thousand monosaccharide subunits. They are non-crystalline, which means now if you take simple sugars, normal the sugar which we find at our home, right? Uh, those sugars have a crystalline shape, right? But these polysaccharides are not crystalline shape. That is the thing. Water insoluble. The normal sugar we find at our home, which is glucose, if we take the normal sugar and if we dissolve it in water, what happens? It will be completely dissolving, form a homogeneous mixture. But if we dissolve a polysaccharide, they won't dissolve itself, which means they are water insoluble, right? So last one, therefore, they are not considered as sugars. These are the three features coming under polysaccharide, right? After this, I introduced you this diagram, which means polysaccharides are classified into two, right? Based on their function and form, right? They're based on their function and form. If you see function, they are used in two function. What? First one, they are used in structural purpose. Polysaccharides are finding in structural function. They have structural contribution. Second one, they have a storage component, which means polysaccharides are used to appear as a structural component, and second one, they are used as storage components. If you see structural, cellulose, just cellulose, right? Cellulose are found in plant cell wall. Now plant cell walls gives the plant cell a correct shape, like a frame. So they are having a structural component. If you see storage, glycogen is present in animals. They are the ones that are storing glucose in animals, right? These are the examples. Now you might ask, sir, on here we know cellulose, we know glycogen, we know starch, but we are not used to these words. Hemicellulose, pectin. What are they? Right? We don't worry. We'll be learning about them separately. Here, see, there's a table. We'll be learning about them separately. Now, for the time being, I want the classification to be clear, right? If you see form is separated. They have two forms. Polysaccharides have two forms. First one, linear, branched. Simply they appear as linear or branched, right? Linear example, cellulose is a linear polysaccharide, amylose is a linear one. Branched means hemicellulose is branched, amylopectin is branched, glycogen is branched. Is that clear? Now this is the thing. Simply the classification part. Is that clear? How polysaccharides are classified? If you still can't understand, let me know. I'll explain it again. Right? No, even if you now understand, if you can't understand even now, it's not your mistake. It's my mistake because I am not, trying to explain it properly. So don't hesitate to ask it again. If you can't understand, I'll explain it. Understand, right? Understood, right? Clear, right? Is it clear for all? I hope it's clear, right? All right. Let's move on back to the note part, right? That is the thing they are saying. Some polysaccharides act as storage components where others contribute to structure. Based on their function, they are categorized into storage and structural polysaccharide. Example for storage, starch and glycogen. Let's see. Yes, starch and glycogen. Right? Structure, cellulose, hemicellulose, pectin. Let's see. Cellulose, hemicellulose, pectin. Correct, right? That's the thing. Based on the architecture, they are categorized into linear form. Example, cellulose, amylose. Let's see. Correct. Cellulose, hemicellulose. Right? Branched forms, glycogen, amylopectin, hemicellulose. Let's see. Like hemicellulose, amylopectin, glycogen. Correct, right? Now, here they are given it in four different points, right? Three different points, right? Three different points. I have given simply in a diagram. That is the thing, right? That is the difference, right? Wait a minute. Right now, we are coming on to the examples of polysaccharide. Let's see. Starch. Now, this is not a new word to you. Starch we have learned in our O levels. Starch is made up of monomer. Is the word monomer new to you? First, tell that. Is the word monomer new to you? If it is new, just drop a message. Feel free, right? Feel free to ask doubts. That's what I want, right? Don't think sir will think like this, sir will think like that, right? No, I won't be thinking anything. If you don't understand even single word, just let me know, right? I'll explain that. Do you know the word monomer? What is monomer? Yes or no? No? I'll explain. Right? Now this is the thing. Now if you see polymer, polymer means A collection of, yeah, no, simply a collection of molecule. Now just simply say there is a classroom. No, now let's say this is a room, right? This is a room. Now this room was built by bricks. Am I right? Now multiple bricks, right? There are four walls and each wall has bricks in it, right? Now can you understand that room is a polymer? The bricks are monomers. Monomers means the building units. Can you see? Now can you understand what is monomer mean? Can you understand or not? Yes or no? Students, students, can't you hear me? Is that clear? No, it's really hard. Yes, clear, right? I hope it's clear. Is it clear for all? Please drop a message quickly. Yes or no, that's it. Don't think about others. If it's clear for you, just drop a message. Yes, sir. If it's not clear for you, just drop a message. No, sir, I didn't get it. That's it. Yes or no? Is it clear for all? Yes, right. Perfect. Hmm. Right. Okay. Now, if you see, starch monomer is glucose, which means glucose combines to form starch. A collection of glucose will be combining, right, forming a starch. Right. Right. Let's see the function of starch. Starch is found, it is storing glucose in plants. Now, this is the thing. Now, in plants, starch stores glucose in plants. Can you see? Starch stores glucose in now. Now you're just getting used to this, right? Now, what you do is, if you have the now, do you have the tute as a PDF, or do you have the tute as a printed copy? First, tell me that. Right, if you have a small notebook, or if you even if it is a resource book, no issue. What you do is, now, just for the explanation, what you do is, just above this part, write, now write like this: starch stores. Now they have just mentioned that stores, it is functioning. Now see, a function stored in plants. Now we can understand what does it store. Starch stores glucose in plants. There is actual meaning. If you want, just note it down. So if you go further, you can understand that glycogen monomer is glucose stored, which means glycogen stores glucose in animals and fungi. That's the thing, right? Just drop a message. Is it clear? Students, is it clear? Mm. So it's clear, right? Okay. Have you noted down that? Just write it down. Starch stores glucose in plants. That's it. Right? Yes. Mm. Now we are coming to glycogen. Right? Glycogen monomer is glucose, which means it stores glucose in plants, it stores glucose in animals and fungi. Right? Cellulose. Cellulose monomer is what is the monomer? Glucose. Component of cell wall. Now you can't use that. Right? Now you can't use that. Why? Because Now, if you see starch and glycogen come under what type of polysaccharide? Tell me that. What type of polysaccharide? See here, starch and glycogen comes under what type of polysaccharide? Come on, come on. What type of polysaccharide? Just see the diagram. What type of polysaccharide? Students? Storage, exactly. Very good. No, quickly respond. That's it. You know the answer, right? Don't hesitate, right? Okay. Storage, right? Only for storage we can use that. It stores glucose. But now if you see cellulose, cellulose is what type of polymer? Cellulose is what type of polymer? See the diagram, cellulose. What type of polymer? Oh, what type of polysaccharide? Structural polysaccharide, exactly, right? So for structural polysaccharide, we can't use it because structures don't store, right? Structural polysaccharide does not store. So cellulose is made up of a monomer which is glucose and it is found as a component of cell wall. Cell wall like cellulose theorem, right? It is a component of cell wall. That's it, right? Pectin. Let's come on to the pectin. Pectin is made up of a monomer called galacturonic acid, right? Galacturonic acid. This is a new type of monomer here, right? Galacturonic acid, and is found as a compound of middle lamella. So what is this middle lamella, right? So wait, right? So what is this middle lamella? You might ask, so what is this middle lamella? Now this is the thing. Now if you see, now you will be learning this in your upcoming lessons. Now this is the thing. If you see the plant cell wall, the plant cell looks like this, right? Right. Now inside the plant cell there is a plasma membrane. Am I right? There's a plasma membrane. But there's an untold thing. If you see, this is the cell, right? Now see this part, right? See, wait, I'll give another color. Right, this white part, that is the thing that comes under cell wall. Am I right? Right. If you see the red part, that is the component or that is the thing which is called the plasma membrane. Right? Can you understand that in a cell? Yes or no? Girls, can you agree with that? Don't also just draw another diagram and explain it. I can't understand. Tell it again. Okay. Now this is the thing. Now you can remember plant cell, right? Plant cell. Can you remember plant cell that in your old was there was a box shaped structure which they give, right? So they are. Can you recall that? Now there is a cell wall. This just think this white part as the cell wall and the red part as plasma membrane. Can you agree with that? This is how it looks like, right? Cell wall is the outermost component, and the innermost component is the plasma membrane. Is that clear? Just inside. Ah, yes, very good. Now, but now we learned this cell wall. Now, how we learned was it is just looking like a cell wall, a strip of cell wall. But actually, it is not looking like that. That is the truth. If you ask how it looks like, now this is the thing. The cell wall also is separated into two, right? Now we'll be learning this separately, but for the time being, I'll explain like this. The outermost part now it separates into two, right? Wait, wait. There's a delay. Do you find any delay? Now, see here. become bit struck something when I'm writing or when I'm erasing. Is there any delay? Yes or no? Do you find it as a flow or just there's a delay in writing? Fine, right? I hope it's fine. Right? Wait. Right. You see this as the cell wall. Right, okay. Now this is the cell wall, right? This cell wall is also separated into two. Now that is the truth, right? There's a compound like this, which is the outermost part, right? The blue part, right? It is looking like this, right? What is this blue part called? It is called the primary cell wall, right? Now, the inner part of the cell wall, the green part, this green part is not a single layer. It is made up of several layers. And that one is called secondary cell wall, right? Now, can you see, understand primary cell wall and secondary cell wall combined to form a cell wall? Can you see? Is that clear? Primary cell wall plus secondary cell wall is called a cell wall. The plasma membrane looks like this. It will be looking like this. The red part is the plasma membrane. Mhm. Is that clear? If you can't understand, just let me know. So I didn't get it. Please explain it again. Students, clear, right? I hope it's clear. Is it clear for all? Is it clear for all? Yes, very good. Right. Now this is one cell. Right. Now I'm taking another cell. Now if you take another cell, the gluing agent. See the word I'm using. The gluing agent, which means may cell देकर attach करने के ना, right? Which joins the cell. This is called a gluing agent. Glue means glue. Normally the glue we use to paste something. This gluing agent is called middle lamella. Can you understand what is middle lamella now? Between two cells, the gluing agent that attaches the cell walls of two cells. Can you understand? A glue, it is looking like a glue, a gum. Can you understand what is middle lamella? Yes or no? Mhm. Yes. Is it clear for all? If you didn't get it, just drop me a message. So I didn't get it. Is it clear or no? What is middle lamella? You should understand what is middle lamella, right? Students, Is it clear for all? Yes, very good. Right. Now let's go back to the note. Right. Right. Pectin made up of a monomer called galacturonic acid. It is a compound of middle lamella. Pectin is present. Now if you see this middle lamella, what does this middle lamella actually mean? Middle lamella is a polysaccharide. Can you understand? Middle lamella is a polysaccharide and it is containing, right? This middle lamella contains pectin. Can you see? Now, normally this, now see, this is the thing. Now, middle lamella, wait. I hope this will sometimes confuse you. Now, this is the thing. This middle lamella is a structural component, right? This middle lamella contains a polysaccharide. What is that polysaccharide? Pectin, right? This pectin is a structural polysaccharide which is present in this middle lamella. Can you understand that? Middle lamella is a location. It is a gluing agent that attaches both the cells. In this middle lamella, a polysaccharide called pectin is located. Is that clear? Is that clear to you? Yes. Is it clear for all? Yes, very good. Right. Then let's go to the next part. Right. Pectin is done. Now we are coming to inulin. Inulin is made up of a monomer which is called fructose and it is stored in the tubers of dahlia. Tubers means I hope you have come across tubers, right? In your O levels under reproduction lesson, you came across tubers, right? Normally, if you take the plants contain the roots like this part which stores materials, right? Those are called tubers, right? Those tubers, in that tubers, inulin is present, it seems. It is stored. Now if you see tubers, if you take those tubers, in the tubers, now this is the thing. Now this is now I'll give that responsibility to you. Now I gave an explanation for these, right? How I said that starch stores glucose in plants, right? Glycogen stores glucose in animals and fungi. Now you tell me for inulin, how will it come? Explain that to me. Now I am the student, right? Now explain that to me. You can just try, give a try. How will it come? Friends, just give a try. If it's wrong, no issue. Now say starch stores glucose in plants. Glycogen stores glucose in animals and fungi. Now, similar to that, to explain what is the function of inulin. Just give a try. Don't think will it be correct or wrong. Can you understand what I am trying to? What's my question is actually, students? Now, if you quickly respond, I can quickly finish this and I can go to lipids part, a new topic today. One student has responded. Inulin stores fructose in tubers. Very good, very good. Right, very good. You're right. Very good. The other students also respond. Where stores in tubules of daily. Correct, but you have missed the uh, two main key points. You should the inulin fructose. So inulin stores fructose and you have got the idea, but the fast of typing, right, you have missed those two words, right? So make sure to write that also, right? Very good. You guys have got those idea, right? So don't hesitate to answer, right? Okay, then you have explained the inunion to me, so I hope it's clear. Is that clear? The inunion part, is that clear? The inunion part. Yes, very good, very good, right? Now we are coming to hemicellulose, right? Now don't confuse hemicellulose with cellulose. Hemicellulose is separate, cellulose is separate, right? Right. Now hemicellulose monomer is pentoses and hexoses. Examples for hexoses? Can you remember any examples? Tell me three examples for hexoses. Now, if you truly have revised, you can tell the examples for hexoses without even looking at the resource book. Examples for hexoses. Hex means six. Am I right? 6. N equals 6. Can you remember? Give me three examples for hexoses. Come on, come on. Students? Hexoses. Students? Just give me three examples for hexoses. What are they? Glucose, galactose, fructose. Examples for pentoses, ribulose, ribose, deoxyribose. That's it. Right? So these are the sugars that can contribute in formation of hemicellulose. Right? Right. Now these are found in compound of plant cell wall. See here. I'll take the cell wall. In the cell wall, this part, hemicellulose can be located. They are present as a compound in the cell wall. That's it. Right. Last one, chitin. Right. Chitin monomer is glucosamine. Glucosamine. Function, it is a compound of fungal cell wall. Right. I hope you have have you learned about fungi in your O levels? Fungi. I can't remember because I have, you know, uh, I did O levels almost 5 to 6 years ago, 5 years, I guess. Right? You came across fungi in O levels, right? Right. Fungi cell wall is made up of chitin. Have you learned this in your O levels? Some say chitin, some say chitin, right? Now, in paper, they won't be asking orally, right? So you can write it in anyway, but the spelling should be correct, right? Yes, right. It is compound of fungal cell wall. The fungal cell wall, that cell wall is made up of chitin and exoskeleton of arthropod. I hope this was also exoskeleton of arthropod. This word is not new to you all. You have come across this in your O levels. Can you remember? Exoskeleton. If you can't remember, can remember. Can or can't? Can. Right, very good. Right, that exoskeleton of arthropod also contain chitin, it seems. That's it. Right. So what? Now, normally in A levels, they have asked these questions. They have asked uh, in structure paper. Normally, the structure paper they used to ask what is the monomer of pectin, right? They used to ask, name the polysaccharide that is a component of middle lamella of plants. They can take it in any way, but you should be thorough in this table. Now, don't worry. As soon as we study today, you can't memorize it, right? So with flow, what happens is we'll do papers, we'll do questions, right? Now after completing this portion, I'll be giving you question set, right? We'll practice them. We'll do MCQs, we'll do structure questions, we'll do essays, right? And we'll be keeping on that practice. Hopefully today after completing polysaccharides, I'll be giving an essay to you. Alright, for this week as a homework, I'll give an essay. Alright, by next week, physical students give me the essay as a handwritten. You just write it right after you have 1 week of time. I am not giving, I'll be giving today, example. Now if I complete today, hopefully I'll give the essay by tomorrow, right? I'll post it in the group. uh, I'm not expecting, you to get the, do the questions and right. I'm not expecting you to uh, write it by tomorrow, right? Tomorrow, day after tomorrow. So physical study, what you do, you can give it physically when we meet on next week Friday, right? You just write after revising the carbohydrate portion. You can write the essay, right? And you just bring the essay and give to me. I'll correct it. I'll give it during the class hours itself. So online students, what can do is you have two options. First one, you can write the essay and take a photograph and send me, right? I'll just look at those and I'll make the correction and send you. Or you can request me for an answer scheme. That's your choice, right? Right? It's your preference, right? So slowly, let's start our essay practice also. I hope you haven't started any essays. You haven't. I hope you haven't written any essays in your levels, right? So slowly, we'll start the practice in essays in A levels. I'll explain and I'll teach you how to write essays in A levels. It's not like in O levels, right? You O levels might have written long essays, right? In your first language Sinhala, first language Tamil, you used to write a long essays. Now here we can't write long essays and waste our time. Here we should write the on point information. If they ask a specific information, we should write only about them. We can't just keep on decorating this thing, right? I'll all teach about them, right? Okay. Just check whether this table is clear to you. This table. If it's clear, drop a message, sir. It's clear. Is it clear? Just go through it. So hopefully, let's finish lipids today. By today, I'll try to finish it. So it will be a completion of a molecule, right? Organic molecule. Yes, clear, right? Is it clear for all? Clear. Very good. Right. Now that's it. Theory is done. Right. Now we are coming to carbohydrates of the functions of carbohydrates. That's it. Right. Let's see. Functions. Now they are giving the functions of monosaccharide. First one, energy source. Now this you have learned from your O levels. I hope you have learned. Now if you have done health science in your O levels, you have learned this from your grade 6 onwards. Right. How did you learn about that? You are learning it in carbohydrate. What is the function of carbohydrate? It gives us energy. Simply just remember glucose. In our sports weight, why we are consuming glucose? Why we are having glucose? Because it's a source of energy. Am I right? It is a source of energy. It gives us energy. Right? So I hope this first point it's not clear. It's not that much complex to you. Right? I hope it's clear. Right? Let's go to the second point. Second point is wait. Right. Second point is it acts as or building blocks of disaccharides, polysaccharides. Disaccharides such as maltose, sucrose, polysaccharides such as starch, glycogen. Now relate this with this, right? Building blocks. Normally, if you see, they are the primary or the smallest unit that combine to form disaccharides and polysaccharides. That is what they're saying. Building blocks of disaccharides or polysaccharides. Just let's see the information that they have given under the bracket. They're saying maltose, sucrose, maltose and sucrose. If you see maltose, maltose is formed. Let's see here. See here. The maltose is formed by the combination of two glucose. So this is a monosaccharide. This is a monosaccharide. So two monosaccharides combine to form a disaccharide. So they are building blocks, which means they are the primary units, the smallest unit. Right? If you see polysaccharide, starch. Starch is made up of glucose. Glycogen. Glycogen is made up of glucose. Give me another example. Another example where we can use uh, we can say that those are examples for uh, polymers, or building blocks of they function as building blocks for polysaccharides. Example, cellulose, right? Cellulose is also an example. Why? The monomer of cellulose is also glucose, which is a monosaccharide, right? Fructose also, pentose, hexose, this is also, right? Last one, component of nucleotide. I hope this is the only point you are a bit, uh, it's confusing you. Uh, right. Let's see. I hope it's not that much of hard to understand, but let's see. Now, first you should answer this. In O levels, have you come across this diagram? I hope you have come across at least once this diagram. No, I hope you have come across. Now, if you can't remember, that's a different story. What I want is, have you come across this diagram? I'll I'll explain it. What is those? What is you have come across that? Yes, yes, right, right. Now this is the thing. This circle, this circle denotes phosphate group, right? This pentagonal shape denotes pentose sugar, right? This rectangle denotes nitrogenous base. Now, sir, what are these? Now we can't remember these. What are these? Now this is called a nucleotide. Nucleotide. You might ask, sir, what is a nucleotide? Nucleotide means nucleotides. And now see, what is a glucose? Now if you see here, glucose is a monomer. Monomer of starch, glycogen, cellulose. Am I right? Am I right? Glucose considered to be a monomer, right? Monomer means they are the simplest units. They are the first step, right? They combine and they form a polysaccharide, right? Similar to that, have you heard something like this before in your life? DNA, RNA, right? DNA, RNA, have you come across this? Yes. Now these are, the full form is deoxyribonucleic acid. Let's see. I'll write that. Deoxyribonucleic acid. This is ribonucleic acid, right? Now, right, this is the thing. Now, this is a nucleic acid, right? This is a biopolymer. Biopolymer. Now polysaccharide is a biopolymer, which means it is a polymer, a biological polymer. So, similar to that, this is DNA, RNA is also considered to be a biopolymer. Can you understand that? Polysaccharide, nucleic acid, these are biopolymers. Biopolymers is simply biological polymers, not the chemical chemistry you have learned alkane, alkene, alkynes, right? So that is chemistry, right? Now in biology, these are biological polymers. Can you understand that? which is they are present in the living organisms. Can you understand that? Yes. Now, these are biopolymers. So, what are the monomers of them? Now, if you see cellulose, cellulose is a biopolymer, right? They are polymer. What is the monomer of cellulose? Cellulose is a polymer. What is the monomer of cellulose? Students, what is the monomer? See here, what is the monomer? Students, now keep your energy till the end. So it's just a 2.5 hour class, I guess. So keep your energy till the end. Glucose, right? Okay, glucose. So for cellulose, the monomer is glucose. For DNA and RNA, the monomer or the for the nucleic acids, the monomer are nucleotides. Can you understand? Nucleic acid, it is a polymer, right? Monomer of it is nucleotide, right? Can you understand that? This DNA, RNA are made up of monomers called nucleotides. Right, they differ. I'll explain now. We'll be going in deeper in nucleic acid part. Right now, for the time being, I have to explain that. Is that clear? Nucleic acids are polymers. Nucleotides are the monomers of those polymers. Is it clear? Yes. Is it clear for all? Yes. Right. The only difference here is there's a pentose sugar here, right? Is this the first time you're coming across the word pentose sugar? Pentose sugar is the first time. Is this the first time? No, we have come across it in A-levels also, right? Pentose means n equals what number? Pentose means n equals pent means 5, right? 5, right? Very good, right? Now, if the monomer, now if you see, if you take DNA, the monomer of it is a nucleotide, right? That nucleotide would contain what type of pentose sugar? Now, this is a bit hard, not hard now. Now, you are just coming after all those, right? If you guess this correctly, you have got the concept exactly. Nothing to explain. Now, see the word, will you? DNA. D means deoxyribo, N means nucleic, A means acid, right? Now, we'll consider DNA. This is a polymer. For DNA, the nucleotide will look like this, right? Right, it will be looking like this. For the pentose sugar, what will be the pentose sugar? Let's go back to the pentose sugar part in our A level. Right now we have learned about pentose sugars, right? Let's go back. Right, here the pentose sugar. For DNA, among these three, what can be the pentose sugar? Just give a guess. For DNA, from these three, the pentose sugar can be ribose, deoxyribose, or ribulose. Why? Just, I said to guess, yeah, but that guessing also should have some sort of reason, right? Deoxyribose, exactly. Right? That is what I said to see the name. Now, see here, DNA means, what is the meaning of DNA? Deoxyribonucleic acid, right? So, deoxy. Now, here, deoxy means what they are trying to convey. The sugar should be deoxyribose. Can you understand? Now they are not giving the full term. They are indirectly saying deoxy means they are denoting deoxyribose sugar. Can you understand? Yes or no? Can, right? Very good. Right. Now I'll give another activity. Now just guess for RNA. See the name and just guess for RNA from these three. Ribose, deoxyribose or ribulose. Ribose. Very good. You have got it. Right? That's the thing. Now can you understand? So if you see DNA, in DNA, in place of this pentose sugar, what will be there? Deoxyribose sugar. Right? For RNA, it will be containing ribose sugar. That is the difference. Can you see the difference? So between DNA and RNA, we have learned the first difference. The first difference is the pentose sugar. In DNA, it is deoxyribose sugar. In RNA, it is ribose sugar. Right? Now, can you see? Now, this is a nucleic acid part. But this nucleic acid part also has a contribution from monosaccharide. Where does it have the contribution of monosaccharide? Where? Now, this is a nucleic acid part. Now, we'll be learning in our, this tute, we'll be learning about carbohydrate, lipids, protein, nucleic acid. Nucleic acid is the fourth topic, right? Now they have just as the functions have included about nucleic acid. I just introduced, right? Now, even if this is a nucleotide, right? Still, this nucleotide also has a contribution of monosaccharide, right? Where does it have a contribution? කොතනද මොනොසැකරයිඩ් ගැන අපි කතා කරන්නේ මෙතන? Where? Just see, just try, I guess. Now, where in this is a nucleotide. Here, this is the structure, right? Here, where have we spoken about monosaccharides? Phosphate group, pentose sugar, nitrogen base. Now, if you can't keep on, dropping the message in chat, you can use your mic also, right? Why? I want a proper reason for your guessing. Right. Both of you just drop a message. Where? In this nucleotide, where does monosaccharide contribute? කොතනද මොනොසැකරයිඩ් ගැන අපි කතා කරන්නේ මෙතන නියුක්ලියෝටයිඩ් එකේ ෆොස්ෆේට් ගෲප් එකේද පෙන්ටෝස් ෂුගර් එකේද නයිට්‍රජනස් බේස් එකේද now both of you are saying phosphate. Right, let's see. Now this is the thing. Now see about phosphate group. Now see the pentose sugar. Right? Yeah, let's see. Pentose sugar. Now if you see pentose sugar, does it a monosaccharide or not? Is pentose sugar a monosaccharide or not? See here. Is pentose sugar a monosaccharide or not?
Yes, monosaccharide.
Yes, it is a monosaccharide. Right? Right? Now this is the thing. Glucose is not present. Right? Pentose, what are the example? Ribose, deoxyribose, ribulose. Right? These are the examples. Now, what was my question? Now, see, these three are the examples for pentoses. So, pentoses means simply this is the formula. The N value, if the N value is 5, they are considered to be pentose sugars, right? Now, what was my question was, now I said that nucleic acids are biopolymers, right? These polymers are made up of some monomers called nucleotide. The structure of nucleotide is here, right? The structure, now this you have might have learned in your O levels, right? This is phosphate group. The circle denotes phosphate group. The pentagonal shape denotes pentose sugar, nitrogenous base, right? Here, I say the question I asked was, where does monosaccharide contribute in this nucleotide? In the name itself, it is denoted. See, pentose sugar. Pentose sugar is a monosaccharide, right? Can you see? Pentose sugar is a monosaccharide. If you want example, I explain that also. See here, DNA, RNA. These are nucleic acids, right? Now, don't think a lot, sir. I don't understand nucleic acid. Now you are just going on to nucleotides. You're connecting those and these. Don't worry, right? Now, what is our primary motive? What is our actual idea? Now I want you want to understand only monosaccharide. I want to explain the monosaccharides contribution here. Don't consider this what is DNA, what is RNA, those stuff, right? Just simply understand DNA, RNA are nucleic acid. If you can't you can't remember as nucleic acid, just think they are biopolymers. They are polymers. Just think as polymers. These are polymers. If you see DNA, this full term is deoxyribonucleic acid. If you see RNA, it is ribonucleic acid, right? Okay. Now just consider, now this is the nucleic acid. If you see the, now this is the nucleic acid, right? Its monomer is a nucleotide. If you consider the monomer of DNA, here in this pentose sugar place, what will be the sugar? By looking at the name itself, just let me know. Now DNA, which means deoxyribonucleic acid. If we consider the nucleotide of this DNA, the pentose sugar, let's go to this back to again. In this pentose sugar, what we have? Ribose, deoxyribose, ribulose. What will be the sugar?
Deoxyribose.
Deoxyribose, that's it. Can you see? Now let me know for RNA. RNA means ribose nucleic acid. Ribonucleic acid. What will be the sugar? Pentose sugar for it.
Ribose.
Ribose, that's it. Now can you see the pentose sugar is the one that is contributing in nucleotides, right? So pentose sugar is a monosaccharide. So there's an indirect contribution of monosaccharides. Now let's go to the function. Let's see the function. See here. Component of nucleotide. Will you agree with me now?
Yes.
Yes. Is it clear for all?
Yes, sir.
Right? Is it clear for all? Because now say, wait. Yes, is it clear, right? Is it clear for all?
Yes, sir.
Yes. Right. Very good. Very good. Right. Okay. Right. Now we are coming to the end of monosaccharide. Now monosaccharide, that's it. End. We have finished the theory. We have finished the function part. Now we are going on to the disaccharides. Right. Right. We are moving on to the disaccharide part. If you finish disaccharide and polysaccharide, we are coming to lipids. Right. Disaccharide. Function, storage sugar in milk. Right. Now we can't read this directly like this. Now if you read just like this, we can understand actually what they are trying to convey. Right. Now see, storage sugar in milk, lactose. What is this? Now we should understand like this. If you have a notebook, if you have a resource book, if you have the tute of mine, you can write it anywhere, right? What I want is you want to understand what the function means actually, right? Now this is the thing. Storage sugar in milk means lactose is a disaccharide. So this is the thing. Lactose stores sugar in milk. Can you understand that? This should be read like this. Lactose is sugar, right? Which is the one that that stores the sugar in milk. Just because of lactose, sugar is present in milk. Can you understand that? If you can't understand, drop a message. Or you can use the mic also.
Understood.
Clear. Very good. Second one is translocation in phloem. Now this is the thing. Translocation of phloem. Now we'll be learning this in unit number 4, which is under plants. Now this is the thing. Simply, we know about phloem tissue, right? You might have learned it is a type of vascular tissue. What is the function of phloem? Phloem involves in transportation of nutrients to the plant, right? Nutrients. If you see, this is the soil. This is the plant. There is roots, right? From the soil, nutrients are transported into the root. From root, there are vascular tissues that are present here. Through the vascular tissues, they are transported towards the upper parts of the plant body. So, if there are nutrition, what is the vascular tissue that contributes? Xylem or phloem for nutrition movement. Phloem for water, it is xylem, right? So, the sugar, now see sucrose. Now, translocation in phloem is sucrose is the one that is helping in movement of phloem. Now this is the thing. Now how actually it looks like means now this is the thing. Uh, let's take like this, right? Right. There is a location. Now normally this will be taught to you in your unit number 4, right? Now just stay. This is the location where sucrose is produced in a plant. Can you understand? This is the location where this is the location where tell me. It is the location where sucrose is produced in the plant. Just for an explanation. Now, translocation of phloem means now this is the thing. Now this sucrose should be transferred. Now in human body, how it is transferred through the blood? It is what is the sugar that is transferred through the blood or nutrition? Glucose. Am I right? Even if in blood they'll be checking the glucose, right? Yes or no?
Yes.
Yes, sir.
Yes. For plants, if we consider the plants, we are using the word sucrose. Can you understand how? What is the difference? For in simple difference is in humans it is a monosaccharide, in plants it is a disaccharide. That's the thing, simple idea, right? Okay. Now this sucrose that is produced in location should be transferred to the relevant location, right? For the location where they should be transferred actually, right? This transferring process is called phloem translocation. Is that clear? Whereas, sir, why have they given the word phloem translocation? It should be sucrose, right? This transferring is done through the tissue called phloem. Can you understand? Through the vascular tissue called phloem. As this translocation takes place through phloem, they are saying phloem translocation. Is that clear?
Yes, sir. Clear.
Yes, very good, right? Clear, right? Okay. Now, during this phloem translocation, what is the sugar that is converted, or what is the sugar that is carried through phloem?
Sucrose.
Sucrose. That is the simple idea. Just now try to understand translocation in phloem, sucrose. Can you understand that?
Yes, sir.
Can right? Very good. Okay, last one. Stores sugar in sugarcane. Now, this is the thing. Sucrose is the thing that is present in sugarcane, right? Due to the presence of sucrose will be storing. Now this is the thing. Let's see. Glucose plus fructose forms what sugar?
Sucrose.
Sucrose. Very good. Sucrose, right? Now They are saying sucrose is the one that stores sugar in sugarcane, right? Sugar means sucrose is present. Sucrose is a disaccharide. Now, if the plant sugarcane contains sucrose, this sucrose can now you should have a knowledge, three-dimensional knowledge. Now, see, sucrose is present. Now, what is this box? This is sucrose, right? They are saying sucrose stores sugar. Can you understand this sucrose contain glucose in it? Yes or no?
Yes, sir.
Yes or no?
Yes, sir.
Yes, right? That is the thing they are saying. Sucrose stores sugar in sugarcane. Storage sugar in sugarcane, which is done by sucrose. Now just check these three functions of disaccharides and let me know if it's clear. If it's clear, I'll move on.
Yes, sir. Clear.
Clear, right? Is it clear for all?
Yes, sir.
Yes, very good. Right? Right. Last function is polysaccharide. Clear, right? Right, very good. Last function for polysaccharides. Now I said that polysaccharides are divided into two based on their function as storage and structural. Let's see the storage. See here, starch. Now can you understand? Now, first initially I said to write like this. See here. Now I said write like this. That is useful. See here, they have written like that. This is the actual meaning. Now see, starch stores glucose as energy source in plants and chlorophytes. Chlorophytes means the organisms that carry photosynthesis. Now, in our all levels, we know that plants are the one that carry photosynthesis. Am I right? Right? Photosynthesis. But now we have to learn that any organism that can have chlorophyll, chloroplasts, they can carry photosynthesis, which they can produce themselves food. Right? Now, if you see green algae is green in color because it has what? Now, plants are green in color. Why? They have which organelle?
Chloroplast.
Chloroplast.
Chloroplast. So, chlorophyll, both of it, right? Ah, chloroplast, mainly organelle means chloroplasts, right? So similarly, green algae is green in color because it has that chloroplast in it, right? Chlorophyll pigments are present. Now we'll be learning them in unit number 3, right? Okay, chlorophytes means simply understand the organisms that carry photosynthesis. Or you can put a bracket and write the organisms that have chloroplast in it. So either way, glycogen stores glucose as energy source in animals and fungi, right? Inuolate stores fructose as energy source in dahlia tubers. These I have explained in the table itself, right? Let's go to the structural polysaccharide. Cellulose in the cell walls of plants and chlorophytes. They are present in the plant's cell wall and chlorophyte cell wall too. Pectin is present in middle lamella of plant tissues, right? I hope you can understand the middle lamella part, right? I explained to you guys, right? I hope it is clear, right? Is it clear for all that part, which is the middle lamella part? Because it is new to you all. You haven't come across middle lamella in your O levels itself, right? I hope a student joined a bit late. I don't know whether she was able to understand that the middle lamella part. Was it clear? The middle lamella part. I don't know whether you joined before that part or after the middle lamella part.
I joined later.
Later.
Yeah.
Now this is ah wait. I'll now this is the thing. Right? Now others are a bit manageable. This middle lamella is the thing that is a bit confusing. Now this is the thing. Pectin is a polysaccharide that is made up of a monomer called galacturonic acid. Right? They are saying these pectins are present or they are components of middle lamella. What is middle lamella? Let's see. Right? Now see, this is the thing. Now can you see the white color? This white color and this white color, right?
Yeah.
White color range is the cell wall. Take this as a plant cell. The white color range is the plant cell wall. The red color denotes the plasma membrane. Can you understand that?
Yes, sir.
Yes. Now within that cell wall, now until O levels, what we learned? We learned that the outermost layer is plant cell wall, and then plasma membrane, and then cytoplasm. That's it. But in our A levels, we'll be learning that even the cell wall is divided. Now, if you see this white color cell wall will be divided into two. The outermost layer is primary cell wall. The innermost layer is secondary cell wall. Right? Now, if you see the cell wall, now this is another cell. This is another plant cell. Now, can you see this yellow color part? The yellow color part is some like a glue, like a gum, right? This gluing agent that attaches the cell wall of two adjacent cells is called middle lamella. This middle lamella contains pectin in it. Pectin is a polysaccharide. Can you understand what is middle lamella?
Yes, sir. Thank you.
Yes. Welcome. Right. Let's move to the polysaccharide function part. Right. Peptidoglycan in cell walls of prokaryotes. This is a new one. Right. Have you come across this peptidoglycan in your levels? No, I can't remember because too long, right?
No, sir.
Uh, no. Peptidoglycan, right? Fine. Now, before going on to peptidoglycan, I'll go to the chitin part and finish it off. Right? Chitin in the cell walls of fungi and exoskeleton of arthropod. It is present in the fungal cell wall and exoskeleton arthropod, which means the outermost skeleton, right, of arthropods. It is located. Peptidoglycan. Now, this is the if you see bacteria, right? Rather than using the word bacteria, we can use domain bacteria. Is the word domain new to you all? No, right? Can you remember the hierarchical sequence? Domain, kingdom, phylum, right? It'll be going on, right? Can you remember that?
Yes, sir.
You can't remember? I don't know. One student has mentioned no. Yeah, you can remember, right? Very good. Right? This domain bacteria contains bacterial species in it, right? That bacterial species, all of them, in their cell wall, they contain this polysaccharide. What is that polysaccharide called? Peptidoglycan. Can you understand that? Similar to our plant cell wall. In our plant cell wall, what is the component? Cellulose is present. Hemicellulose is present. Right? So similar to plant cell wall, in cell walls of domain bacteria, they contain peptidoglycan. But you should be very careful that domain archaea, right? Domain archaea does not have this peptidoglycan in it. Is that clear?
Yes, sir.
Yes. Now, organisms were classified into two. Can you remember prokaryotes, eukaryotes? Right? Prokaryotes, they were classified into two domains: domain bacteria, domain archaea. Eukaryotes, domain eukarya. That's it, right? Now, in prokaryotes, domain bacteria is present, domain archaea is present, right? In these two, for which domain does peptidoglycan is present in the cell wall?
Domain bacteria.
Domain bacteria, exactly, right? Is it present in domain archaea?
No.
No. Now with this idea, go to that. Now this is the problem. See here, they have used the word prokaryotes. Now students might they might confuse, or they can go to misconception that both domain archaea and domain bacteria would contain. No, only domain bacteria contains peptidoglycan. Now just check this and let me know if it's clear. If it's clear, we can move to lipids.
Sir,
Clear? Clear to all?
sir, can you explain the last part? I didn't get it.
Last part means you mean the peptidoglycan or chitin? Peptidoglycan
Peptidoglycan.
or Sorry, the peptidoglycan part or the chitin part?
Peptidoglycan part.
Peptidoglycan, right? Now this is the thing. Now you can now see the plant cell wall. Plant cell wall contains cellulose, right? It contains cellulose, hemicellulose in the cell wall. So similar to that, bacteria, the normal bacteria, their cell wall contains peptidoglycan in them, right? But as they have mentioned it as prokaryote, the problem is students will be going to they'll be misunderstanding that both domain bacteria and domain archaea would contain. No, actually, this is the thing. Let's see. Prokaryotes are divided into two: domain bacteria and domain archaea, right? Here, only domain bacteria contains peptidoglycan in their cell wall. If you see domain archaea, they don't contain peptidoglycan. They contain other components like polysaccharides, proteins, polysaccharides. They have other components. Only domain bacteria or bacterial species contain peptidoglycan in the cell wall. Is that clear?
Yes, sir.
Right. Is it clear for all this part? Now we are coming to the end of the carbohydrate. Right? Right. One student has said that it's clear for her. Is it clear for all?
Yes, sir. Clear.
Right. Very good. Okay, then let's move to a new topic, which is lipids. Right? Okay. Now, before just we are keep on reading the notes. Right now, just for a change, let's keep a small discussion. Right now, as soon as you come across the word lipid, what comes to your mind? Just right, just as come across the word lipids, what does what is your inner feeling try to convey? What actually feels? Lipids means fats, oils, right? Some might feel junk food, right? Lipids, those, right? So lot of things, right? Simply some have a feeling it is unhealthy. Am I right? Some have the
Yes.
inner feeling that lipids are unhealthy, right? But actually lipids are very 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 keen, that was very very important for our human body. But actually we are failing to understand that. We think glucose is good, but we think lipids are bad. Am I right? Right, that is
Yeah.
a normal understanding. But if you see glucose, the priority we give for glucose, equal priority should be given to lipids as well, because both of them are functioning as energy sources. I'll explain how, what is the actual contribution of lipids, right? So here what we'll be learning is, we'll be learning about lipids, the types of lipids, we'll be learning about functions of lipids and structure of lipids. That's it. Simply we'll be learning how lipids are formed also, right? Okay, let's go to the topic. Lipids. Lipids are diverse means more common, right? The word diverse means common group of hydrophobic molecule. Hydrophobic means, are they water loving or water hating molecules?
Water hating.
Water hating molecules, right? Water loving molecules means
I
hydro.
think they dissolve in water or like that.
Yeah, you can use that for also, right? So simply, no, this is the thing. These are Greek words, Latin words, right? Now, if you see the word philic actually means loving, right? But the actual meaning hydrophilic means they react with water. As you said, that is the actual idea. Hydrophobic means they don't react with water. That is the thing. But now, as directly without going on to the dissolving part, I just went with the word. Philic means loving. Phobic means hating. That is the thing idea. right? Is that clear? Is that clear?
Yes, sir.
Yes. Now simply hydrophobic molecule means there are two features. First one, they don't react or they don't dissolve in water completely or in water, H2O, right? Second, they completely hate water, right? Now actual scientific idea is this. Now this you can understand for your understanding purpose, you can use that, right? Okay, second point. They are large biological molecules but not considered as polymers. This is very, very important because this has questioned in your past papers also. Students confuse lipids with macromolecules and biopolymers. They are large biological molecules but not polymers or macropolymers, macromolecules. Can you understand that? Lipids are large molecules, but they are not polymers, which means they are not formed as a chain reaction or they are not formed by small, small monomers. They are large molecules. Naturally, they are large molecules. They are not formed using monomers. Can you understand that, students?
Yes, sir.
Yes. Okay. Yes. Third one consists of carbon, hydrogen, oxygen. Right? Carbon, hydrogen, oxygen. Now you should be very keen in this because see here. Now carbohydrate. While we were learning carbohydrate, can you remember the elemental composition? See here. What is the elemental composition? See here. Carbon, hydrogen, oxygen. Right? Now we are in lipids. Lipids also carbon, hydrogen, oxygen. Right? When you are going to proteins, there will be an addition. When you are going to nucleic acids, there will be addition. Right? So don't confuse that. So let's see what are the these are mainly elemental composition, right? And the H is to O ratio is not 2 is to 1, right? Name a compound which has H is to O ratio 2 is to 1, which means if you consider that molecules H is to O ratio, it should be 2 is to 1. Name a molecule.
Water molecule,
Yeah, water molecule. Very good.
carbohydrate.
Carbohydrate, exactly. I wanted carbohydrate molecule, right? So water is also correct, but I wanted an organic. The mistake was mine because I didn't use the word organic, right? So carbohydrate molecule is having H2O ratio 2 is to 1. But lipids does not have, you should be very, 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 keen, or you should pay more attention to these words like not, major, large, comparatively, right? These words should be very important because MCQs, they won't be given directly. They'll be jumbling completely. The main idea they want is they want to check the students utmost knowledge on that section, so they'll be completely jumbling the words, but you should be having that idea. So see here, first they said that they are large molecules, not polymers or macromolecules, and here they don't have a 2 is to 1 ratio of H is to O, right? Comparatively more hydrogens are present, it seems, right? More than 2, right? Last one, biologically, important types of lipids. Lipids are classified into two: fats, phospholipids, steroids. Is this introduction part clear to you?
Yes, sir.
Right. Is it clear for all? Yes. Right. Okay. Now, under these three, fats, phospholipids, steroid, we are coming out. We are coming across the first topic, which is fats. Right. The normal fats. Let's see. Fats are made up of glycerol and fatty acid. Glycerol, fatty acid. Glycerol belongs to alcohol group having three carbons where each of them bear single hydroxyl group. Now, first I'll read this completely and then I'll explain, right? Fatty acids are hydrocarbon chains with long 16 to 18 carbon skeleton with an carboxyl group at its terminal. Fatty acid molecules bind to each hydroxyl group of glycerol by ester bond, resulting fat molecules called triacylglycerol. Now this is the thing. I'll explain. Right? Right. What is this blue color? OH means hydroxyl. Right? Okay. OH means hydroxyl. Right? Because now you are coming, you're doing A level science, right? Now in your A level chemistry, you will be learning across these words, right? Hydroxyl, cyanide, right? Thiocyanide, sulfate, those words will be learned. Now this is the first one. What is this? Hydroxide, right? Hydroxyl, right? Hydroxyl. Now, what is this molecule? This molecule is the one called glycerol. So glycerol has how many carbons?
Three.
3 carbons. Each carbon is attached to how many hydroxyl groups?
Three.
No, no. Listen to the question carefully.
Two.
Each carbon is bound to how many hydroxyl groups?
One.
1, right? Okay. As each carbon is bound to a hydroxyl group, we say it is an alcohol, right? Now see, have you come across the word ethanol in your levels? Ethanol means it's normal
Yes, sir.
alcohol. Yes. So ethanol has a chemical equation of CH3CH2OH. So as it has an OH group, this is said to be an alcohol. Can you see? So here, how many OH is present? 3 OH. So it should be definitely an alcohol. Is that part clear? This is the glycerol part. Is it clear?
Yes, sir.
Yes. Now let's go to the fatty acid part. Now fatty acid is a organic acid, right? Organic acid means it should contain carbon, hydrogen, right? That should be present. Now if you see carbon dioxide, is it organic or inorganic?
Inorganic.
Inorganic. Why? It does not have hydrogen. If you see water, is it organic or inorganic?
Inorganic.
Inorganic. Why? It does not have carbon in it, right? So fatty acid is an organic acid, which means it has carbon as well as hydrogen in it, right? So, but the problem here is fatty acids are long. Long means it has 16 to 18 carbon. So instead of using it as a long, they are saying it has a long carbon skeleton. It is a long pattern. The pattern of long carbon is called a skeleton. Now it will be looking like this actually. Like this. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15. Let's take 16. Now this it will be looking like this 16 carbon atom or 18, 6 to 18, 16 to 18. It will be varying. And the problem is it will be not ending like this. It will be having a carboxylic terminal at its one end. It will be looking like this. So here it will be having hydrogen. It will be having hydrogen here, right? He'll be having hydrogen here, here hydrogen, here hydrogen. It will be going on. Can you see? Right? It will be completely be having a hydrogen, right? If you want, I'll draw. The problem is the time, right? Here one. Now here, as the paper is white, I'll give this hydrogen a red. Right. Right. What is this complete molecule called? This complete molecule. Just now I said. What is it? The name. Fatty acid. What is it? What is it?
Fatty acid.
Fatty acid. Right. Now fatty acid has a carbon skeleton. Now see this carbon skeleton varying from how many carbon to how many carbon? The range. 16 to 18 carbon. Now they are as this. Now see, this is the part. Wait, I'll take this. This is the part that is called the carbon skeleton or chain. See here they are using the word long carbon skeleton chain, right? Hydrocarbon chain. Hydrocarbon chain, right? Hydrocarbon means it should contain carbon and hydrogen. Can you see? This is a hydrocarbon chain. This red part completely. Now I have excluded this green part. Now green part is also attached at its one terminal, which means at one end it is attached. But I have excluded that. I have said that this part is completely called the hydrocarbon chain. Can you understand that? Will you agree with that?
Yes,
Hydrocarbon
sir.
means it should contain only hydrogen and carbon. Does it have only hydrogen and carbon in this red part? Just look at this red part here.
Yes, sir.
Yes, it has only. Are all the students agree with that? This red part has only carbon and hydrogen, sir. That's it. Will you all agree with that?
Yes, sir.
Yes. Now here, as this hydrocarbon tail or the hydrocarbon chain has only carbon and hydrogen, this is said to be hydrophobic. Why it is said to be hydrophobic? What is the reason? It has only, what are the two elements it has?
Carbon and hydrogen only.
Only carbon and hydrogen. Very good. Only carbon and hydrogen. Right. Okay. This green color part is called. Wait, I'll give another color. Right. This is called carboxyl terminal. What is it? Carboxyl terminal. Right. Now this is also attached at one end. Carboxyl terminal. This carboxyl terminal contains carbon, hydrogen, oxygen. Now you can guess, is it hydrophobic or hydrophilic?
Hydrophilic.
Hydrophilic, very good. Right. Can you see? Now, just think, is it practical for a student to draw this complete structure in a paper? Is it easy? Is it an easy work?
No.
No. So, instead of this complete red part, we use a simple term called a variable. Marasa, what is that variable? Now, this is the thing how it looks like. Now, here I'll draw the carboxyl terminal. This is how it looks like, right? Now, here instead of that complete hydrocarbon chain, we will be writing a letter R. Can you understand? Now, R, there is no element, there is no element called R. So instead of writing a complete chain like this, we used to write a letter R denoting the hydrocarbon chain. Can you understand that?
Yes, sir.
Yes. So this complete fatty acid molecule can be simply written as like this, right? Can you understand? I hope it's clear.
Yes, sir.
Yes. Now, the problem here is, now this is a glycerol molecule, this is a fatty acid. Now, not yet we have come across the word fat. What is fat? Fat is made up of glycerol and fatty acid. These two combines to form a fat. You might ask, how? How does it combine? How it is combining means, this is the thing. I'll write the reaction also, right? Now, to explain the combining reaction, to explain the fat formation reaction, I'll be using this diagram. Is it okay? Can I use this diagram instead of fatty acid?
Yes.
Yes, because this complete diagram I have shortened, right? I have shortened into this diagram, so I can use that also, right? Now what we are going to learn is fat formation. So in our syllabus, for fat we use another word called triglyceride, or we can use triacylglycerol also. Have they have given, right? Triacylglycerol we can use, right? We can use triglyceride also. So as they have given triacylglycerol, I'll use that triacylglycerol. Right. So we are learning formation of fat. Let's see how it is forming. Right. Now I'll draw the glycerol molecule. There are three carbon atoms. Each hydrogen atoms are present, and I have hydroxyl groups. As these have hydroxyl groups, glycerols are considered to be what type of molecules? What type of molecules? If we have OH, it is an, come on. If they have an OH, they are considered to be what type of molecules?
Alcohol.
Alcohols. Very good. Right? Right. Now this is glycerol. Now I'll draw like this. Yeah, this is good. Right? Right. C double bond OH R C double bond OH R C double bond. Right. This is fatty acid. Now you might see, so is this biology or chemistry? Because normal chemistry lessons have this type of reaction. In our syllabus also, there are few occasions where we'll be coming across this type of reactions. Only few, right? Not more than five, right? So no worries. Right. Now, how does the reaction look like? Now, the reaction look like, it will be looking like this. Now, can you see? I'll use yellow color. That's better. Right. There's a OH group here, right? Right. There's a OH group here. There's a OH group here. Now, from your carbohydrate knowledge, can you remember there is something called condensation reaction? Can you remember?
Yes.
Condensation reaction means it removes a water molecule. Removes water molecules. So to remove water molecules, there should be how many hydrogen?
Two hydrogen.
Two hydrogen. How many oxygen?
One.
One oxygen. Now here there are two hydrogen, okay, but there are two oxygen. Which oxygen will choose? That is the problem. Now most of the students, 90% of the students know how to draw this, right? The problem is they don't know how, where to get the oxygen here, right? They confuse and they jumble it. Now we'll be drawing this like this, right? Carefully see. The oxygen from fatty acid, oxygen and hydrogen from fatty acid, and hydrogen from glycerol will be removed. Again, hydroxyl, oxygen and hydrogen from fatty acid, and hydrogen from glycerol will be removed. Again, hydroxyl or OH from fatty acid and hydrogen from glycerol will be removed. Is it clear? Is that clear?
Yes.
Students? Yes. Clear, right? Can you see how it is reacting? OH from this and H from this. Can be removing. Right? Now now these will be removing how many water molecules?
3 water molecules.
3 water molecules. 3H2O. Now let's see how the structure looks like after the reaction. After the reaction, it will be looking like this. Do you agree with this? See here. Hmm. Can you agree with this or not? Do you want an explanation? Is it clear? Is it clear or do you want an explanation?
Clear, sir.
Clear. Is it clear for all? Because this is an important one. Eh? So they have asked this.
Can you explain again?
You want me to okay, no issue. Right? Now this is the thing. Are you clear with this glycerol molecule structure?
Yes, sir.
Yes. Are you clear with this fatty acid molecule structure?
Yes, sir.
Yes. Now, this formation reaction is what type of reaction? It is a condensation reaction. Right? Condensation reaction means it is a water removing reaction. Clear?
Yes, sir.
Yes. For a water to be formed, for water molecule to be formed, how many hydrogen should be present?
Two.
H2O. How many hydrogen?
Two.
2 hydrogen. How many oxygen?
One.
1. So here we have OH, here we have OH. Here have we have, if we see both, we consider both of them, we have 2 oxygen, we have 2 hydrogen, right? But we need only 2 hydrogen and 1 oxygen. Am I right? Am I right?
Yes, sir.
So 1 oxygen will be neglected. So the actual way of drawing is OH from the fatty acid and hydrogen from the glycerol will be removed. OH from fatty acid and hydrogen from glycerol will be removed. OH from fatty acid and hydrogen from glycerol will be removed. So this is a water molecule. This is a water molecule. This is a water molecule. So how many water molecules will be formed? Total. How many water molecules? Here one water molecule, here one water molecule, here one water molecule. How many water molecules will be removed?
Three water molecules.
3 water molecules will be removed. So after removing the 3 water molecules, can you understand this oxygen will be binding with this carbon like this? Can you understand?
Yes, sir.
Is that clear now? Is that clear now? Didn't you understand still? If you want, I'll explain it again. No issue.
I understood, sir.
Okay, right. Understood, right? Very good, very good. Now, this is the thing. Now you might ask, so what is this? A new molecule has formed, right? What is this? This is the one that is called triacyl glycerol. Simply, it is the what? What is that?
Fatty acid.
The fat, the lipid, right? Simply the fat, right? Now let's get back to the note, right? Now there is a problem, not a problem actually. They used to give the marks, but can you agree that if I draw this structure like this, will you agree? See here, both of this diagram. Is it confusing? See here, this carbon has H2, so CH2OH. This carbon has only one hydrogen, so CHOH. This has CH2, so CH2OH. Is it clear?
Yes, sir.
Is it clear? I
Yes, sir.
Yes. All right, very good. That is how they have explained it here, right? Let's go to the note. Fats are made up of glycerol and fatty acids. Now you can agree with that, right? How it is forming. Glycerol belongs to alcohol group. Why? Now you know why it is belonging to alcohol group. Why? Just few minutes ago you said me the answer. Why? Because it has which group?
It has a hydroxyl group,
Very good.
OH group.
Very good. Hydroxyl OH group, right? Fatty acids are hydrocarbon chains. So here, this red color one is called hydrocarbon chain, right? Which has a long 16 to 18 carbon skeleton with a carboxyl group at its one terminal. This green color is the one called carboxyl terminal. Carboxyl means C double bond OH. C double bond OH, right? Fatty acid molecules binds to the each hydroxyl group by ester bond, resulting fat molecules called triacylglycerol. What is this bond called? The bond that joins to this bond. See here. What is this bond called? Ester bond. This is one ester bond. This is another, and this is another. So three ester bonds are formed. Can you see? The bond that forms or joins glycerol fatty acid is called ester. Is that clear? Is that clear?
Yes, sir.
Clear, right? If you find it difficulties, let me know. Don't hesitate to ask. So I didn't get this. I'll explain it again. Right? So with doubts, don't continue. You will be facing many difficulties if you continue with doubts. Is it clear for all? This ester bond.
Yes, sir.
Clear, right? Very good. Okay. Fat molecules. I hope I explained everything, right? Yes. I explained ester bond. Yes. Done. So just check this fat part and let me know if it is clear. Is it clear? Is it clear for you guys? Yes, clear, right? Is it clear for all?
Yes, sir.
Yes, right. Right. Hydrocarbon chains of fatty acid contributes to hydrophobic nature. Can you understand? I said you that this is the hydrocarbon chain. It has only carbon and hydrogen, so it is hydrophobic. Right? Based on the nature of hydrocarbon chain of fatty acids, they are categorized to hydrocarbon chains are classified into two. Right? Into two. Let's see here. Now see. Wait. This is called hydrocarbon chain. So they are classified into two, right? What are they? First one, saturated fatty acid. Right? Now this is fatty acid, right? Saturated fatty acid, unsaturated fatty acid. Now I'll explain what is this. Saturated and unsaturated. Right? Can you understand? Hydrocarbon chain or fatty acid. Fatty acid is classified into two on their nature as saturated and unsaturated. Right? Let's see what are the differences. Now, this is the thing. If you see unsaturated fatty acids, here see this is the hydrocarbon chain, right? In unsaturated fatty acids, here there can be double bonds that are present. Can you understand? Can you understand?
So
Now, I
can
ask,
you
sir,
repeat it again?
I'll explain. No issue. Right now, this is the thing. In unsaturated fatty acids, now this is the fatty acid, right? In unsaturated fatty acids, there can be double bonds seen in the chain here. Now, I'm not speaking about this double bond. This is separate. I am speaking about this red part. In unsaturated fatty acids, that there are double bonds that are seen in this chain. Now, so if you see, these are single bonds, right? One, 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 everything are one, right? In unsaturated fatty acids, there can be double bonds seen. Now you would ask, how? How means like this. Now see, I'll choose this part, right? I'll choose this, right? Wait. I'll draw a double bond here, like this. If I draw a double bond, with your knowledge in O levels, will you agree with me that a hydrogen will be wait. Will you agree with me that a hydrogen will be removed here? Why? Because carbon has how many valency? The valency number is 4.
4.
See, 1, 2, 3, 4. Correct. Can you see? Can you see?
Yes, sir.
Yes. Now you don't go further thinking, sir, why that happens. Now this is the thing. Simply this is the idea. Fatty acids are two. If a fatty now, what is the primary requirement of a fatty acid? Fatty acid should contain hydrogen carbon. Whether it has a double bond or whether it has a single bond, it is a fatty acid. Right? At the end, what is the main requirement? It should react with a fat. It should react with a glycerol. For glycerol binding, does this have a contribution? This fatty acid, this hydrocarbon part, does it have a contribution? This part, the red part. See here, the red part means R. See here, does the R have a contribution here?
No.
No, R does not have. Here, this part is the one that is contributing. So, even if we change the number of hydrogen by creating double bond, will it affect?
No.
There is a small effect, right? There is a small effect. That is a problem, right? Now, see, there are two options. First, there can be double bonds that are present here, right? There can be double bonds that are present here. Second, there can be only single bonds. If there are double bonds that are present in this carbon skeleton, they are called what type of fatty acids? If you have double bonds, unsaturated fatty acid. If you don't have
Saturated.
saturated, that's it. Is that clear to you? I'll explain how the nature changes. Is that clear to you?
Yes, sir.
Yes. Is that clear for all? Is it clear for all? Yes, very good. Right? Okay. Let's go back. Right. See, now I'll write it here. Right. Saturated fatty acid. Right. Saturated fatty acid means no double bond. Right. So unsaturated means have double bond. Not they haven't. There is no compulsory of having only one. So one or more double bonds are present. Right. So will you agree with this point? Now I'm going to tell the next point. Will you agree with this point? See here. The fats. Now this is a fat, right? The fats that are formed from unsaturated fatty acids are called unsaturated fats. Will you agree with that? Now this is the thing. See, there are two types of fats: saturated and unsaturated. Now, I know now this is the first time you are coming across this word side. It is a bit confusing. So now you said there are saturated fatty acid. Now where does this saturated fats come from? Now this is the thing. Now here, if this fatty acid chain contain a double bond, this fatty acid will be said to be an unsaturated fatty acid. If that is the case, if that fatty acid can react with a glycerol molecule and form a fat, right? If it forms a fat, this fat is called a unsaturated fat, right? If a normal one without a double bond, a single bond with single bonds, if it react with a glycerol and form a fat, that is called saturated fat. Is that clear? Simply that is the idea. Is that clear?
Yes, sir. Clear.
If you have any even a minor doubt, just let me know. With doubts, don't continue. Right?
So in that place you have shown a double bond. No, in that place, that H also won't be there. No,
Which
that
H?
second hydrogen you have drawn a double bond.
Yeah, here,
Double
right?
bond. Yeah.
Yeah, yeah. Exactly. It's my mistake. Right. Thank you. Right. Now, is it clear?
Yes, sir. Clear.
Clear. Right. Is it clear for all? I hope it's clear for all. Yes, right? Right. Okay. Now let's go to this point. Right. Wait. Next page, right. Saturated fats. Fats that are made up of saturated fatty acid. Fatty acids with hydrocarbons having no any double bonds. Right? Usually animal fats come under this category. Animal fats does not have any double bonds in their fatty acid structure. So they are mostly solid at room temperature. Example, butter. The normal butter we have, that butter, it has fatty acid in it. What type of fatty acid? Saturated or unsaturated?
Saturated.
Saturated. Saturated fatty acid means, does it have any double bonds?
No.
No. Is that clear? This part. So they have said that they are solid at room temperature. Correct, right? Normally, if you see butter, butter is solid. It is appearing as solid. It is not liquid. It is solid at room temperature. So is this clear? The saturated fats part. Just check and let me know. Clear. One student has said that it's clear. Is it clear for
Ye-
all?
Yes, sir.
Yes. Now we are coming to unsaturated fats. So if similar idea, unsaturated fats means they are made up of unsaturated fatty acid. Fatty acids with hydrocarbons having one or more double bonds. Right? Usually plant fats come under this category. They are liquid at room temperature. Example, vegetable oil. The oil we use to fry something. Right? That oil, it is a fat which is called unsaturated fat because it is made up of what type of fatty acid? What type of fatty acid? The vegetable oil we use.
Unsaturated.
Unsaturated fatty acid. So simply unsaturated fatty acid means it has it has double one or more double bonds, or it does not have any double bond.
One or more double bond.
One or more double bonds. Right? They are normally solid or liquid at room temperature.
A liquid.
Liquid. That's it. Right? These unsaturated fats are further classified into two, depending on their nature of double bonds, as cis and trans. That's it. Now, don't think about what is cis, what is trans, and though, right? They have said it is divided into cis and trans, right? And they have said a small point: consumption of excess saturated fats and trans unsaturated fats contributes in atherosclerosis. Excess saturated fats means, example, butter. Consuming excess amount of butter and trans unsaturated among this cis and trans. If you consume more trans, it can lead to atherosclerosis. Is this the first time you're coming across atherosclerosis?
Yes, sir.
Yes. I'll explain what is atherosclerosis, right? This is the thing. You know our normal blood vessels, right? Blood vessels, the vessels that carry blood. Now it has a wall like this, right? Through this blood comes, right? Moves on, right? Now what happens here is normally when we consume excess amount of fat, specifically what type of fat? Excess amount of saturated fat and trans unsaturated fat. What happens is this blood vessels will be starting to form some cholesterol particles like this. Can you see this yellow color? These are called cholesterol particles. Can you see?
So is cholesterol considered a fat?
Yes. Will you agree with this? Yes or no?
Yes.
Yes. Now it happens like this, right? Now you might ask, what is the problem in this? The problem is, can you understand the rate of blood flow through this starts to increase? Why? Because initially, just think through this 10 milliliters, just I'll just assume 10 milliliters of blood flowed, it was flowing. Now as the lumen or the space, the area decreases, what happens? The volume of blood decreases. When the volume of blood decreases, what happens? Other organs like brain, right, heart, now this will blood vessels will be supplying to various organs, right, those will be starting to affect. Why? The blood rate is decreased. Can you see? The blood rate decreases. So when the blood rate decreases, what happens? Heart start to pump more and more blood. Why? Heart thinks that blood has not reached to other organs, it has blocked in somewhere, I should pump more and more blood, so it starts pumping more and more blood, so our heartbeat starts to raise. So, but the problem is where? In this blood vessels, even you pump more and more blood vessels, here there is a blockage. So blood rate starts to decrease through this. So normally it was 10 milliliters, now it has decreased to 4 milliliters. Even though you pump more and more blood, blood will be flowing only 4 milliliters through this. So heart keeps on pumping, 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 and at a rate, at one instance, the heart starts to, what happened? It will be directly a heart attack will be taking place. Can you understand?
Yes.
Heart will be now, it will be pumping at a very very high rate, and at an instant the heart cannot withhold that speed. So at that instance it can lead to a heart attack also. So can you understand, some most of the people, uh, cholesterol patients, those patients have a high risk of getting heart attack, this just because of this. So their blood vessels can be blocked, so the blood supply can be affected. If the blood supply is affected, what happens? Heart starts to pump more and more blood. With that, their heart gets weakened, pumping more and more blood. Their heart gets weakened. So what happens? Heart slows down. When heart slows down, there is a sudden attack. Can you understand? Can you understand this scenario?
Yes, sir.
Yes. Now you might ask, so is this condition said to be atherosclerosis? Now you are saying about heart attack, right? Now, atherosclerosis means this accumulation of fat in the walls of blood vessels. This accumulation is called atherosclerosis. Can you understand? This deposition of fats and this deposition of cholesterol particle. This is the one that is called atherosclerosis. This atherosclerosis can lead to heart attack. Can you understand that? Yes or no?
Yes, sir.
Yes. Now just go through this part. Is that clear to you all? Now, if just a small part, if you finish that, we can finish lipids within tonight. Only this part, phospholipid part, and then the function. Only few functions. Just go through this part and let me know if there's any doubt. If no doubts, we can continue.
Sir, what is the difference between cis and trans?
Now,
That
cis and trans means this is the thing. In chemistry, you might learn this. Now, this is the thing. I don't know whether is it okay to tell this. In chemistry. You are directly learning this, right? But you will be learning this under organic chemistry, right? Not as soon as you are just now you are introduced into the chemistry part, right? I hope it will be unit number 2 or unit number 1 in your chemistry classes. Organic chemistry comes under unit number 7, unit number 8, those sections, right? So cis and trans means if you see double bond, I'll take like this, right? Now this is a molecule, right? This is a molecule. Now having similar groups in one direction is called cis, which means like this, OH, OH, having H here, having H here. This is cis. This is trans, which means having different groups at one side. Can you understand? This is called cis trans. Can you understand this? Now, this is an example. I explained with respect to fatty acid part. This is example, how it look like.
okay, sir.
Is it clear?
Yes, sir.
Yes. Now I'll explain with respect to the fatty acid, right? Now fatty acid, the problem is I have to draw complete structure. But okay, I'll see how to do it. Now see here. Now this complete structure and this complete structure. Can you see the double bond here? There's a double bond, right? Now I'll draw it like this. Like this. Wait. C double bond C. Now here, see here, here these are the two double bond containing carbon. Here, how many carbons are present on this carbon? 1, 2, 3, 4, 5, 6, 7, 8, 9, and 10. 10 carbons. Here, 1, 2, 3, 4, 5. Here, 5. So, are they similar groups? Here 10 carbon, here 5 carbon.
No.
No. So we are not considering those because they're different. So we are considering only what type of group? Similar type of group. So here what is similar for both carbons? Both
Hydrogen.
this hydrogen. So cis means hydrogens should be in one direction and other group should be in the other direction. So I'm mentioning R1, R2 here, which means these structures are, this is R1 and R2, right? So what about trans? Trans looks like this. Can you understand? It is mainly depending on the orientation. This is called cis trans. Can you understand? Cis means one location, trans means opposite.
Yes, thank you.
Yes. Now it will be too much to you to understand this. Uh, I have explained it also because just because of you asked it, right? Now if you can't understand or if you find it difficult, you can neglect it because they won't be asking those. Draw the structure of cis or trans. They won't be asking because in the syllabus they haven't given, right? They haven't given it, so they won't be asking that. So don't worry. But I'm sure that when you are coming to your A-level chemistry in organic chemistry, you might remember this, right? I'm sure
Okay.
you might remember this, right?
Okay.
Okay, right? Is it clear this complete part? Just check me and let me know. Is it clear? Yes. Is it clear for all? One student has confirmed that it's clear for her. Yes, very good. Right. Okay, then we are coming to second type of fat. Now we second type of lipid. First one was fat. We understood about fats function, about its structure, how it forms, right? What are the types of fats, right? Now we are coming on to phospholipid, right? Let's see. Phospholipids are major components of cell membrane, right? Now, if you see the cell wall, this is the cell wall. Inside the plasma membrane is present, right? That plasma membrane contains phospholipids in it. Can you understand? Can you understand? Plasma membrane contains phospholipids in them. You might ask, sir, how? Now it will be looking like this. Right? It is an interesting part. You might learn in this in the upcoming part. In Unit 2, we have a section called cell biology. There we'll be learning about cell wall, cell membrane. We'll be learning about mitochondria, chloroplast, cytosol. Right? Everything we'll be learning. So there we'll be learning how it looks like. Now, if this is the plasma membrane, it will be looking like this. These are called phospholipid molecules, like this. They'll be looking like this. Right? Can you understand? See here. Phospholipid has a shape like this, right? This is the shape, right? Like this. So this is how it will be looking like. If this is the plasma membrane, no, this is the plasma membrane. These phospholipid molecules will be arranged like this. Here also it will look at it like this, right? Right? Can you understand? Is it clear?
Yes, sir.
Yes. They are composed of two fatty acids, one phosphate group attached to one glycerol. See here, this is the phosphate group. How many fatty acids are attached? One glycerol is attached downwards. See here, the glycerol is attached downwards I'll give the glycerol green color, right? This is the glycerol part. And how many fatty acids are attached? I'll give fatty acids blue. Two fatty acids are attached, right? And there is an additional segment called choline upward. We learn about choline here, right? So simply you should understand that two fatty acids are present, one phosphate group is present attached to one glycerol molecule. Is the second point clear to you? Is the second point clear to you? This is a phospholipid molecule. It contains a choline molecule. It contains a phosphate group. It contains a glycerol molecule. It contains fatty acid. Now, don't confuse this with fat. Now, there we learned. I don't know whether you are confused with that. Now, here, this is different. This is fat. Now, this is fat. Right? See here. We learned that biological import types of lipids. One is fat, one is phospholipid. Now we have completed about fats. Now we are coming on to lipid. So phospholipid, right? We are coming to phospholipid. Lipids can be divided into three: fats, phospholipids, and steroids. Now we have come to phospholipid. This is the shape of phospholipid. Can you understand? Yes. Is it clear for all? One student has mentioned that she's clear with that. I don't know. Clear, right? Very good. We'll go to the third point. The phosphate group gives the negative electric charge to the phosphate molecule, phospholipid molecule. Now, phosphate, if you see chemically, phosphate is looking like this: P O O O PO4 3 minus. Now, this is how phosphate looks like: PO4 3 minus. For phosphorus, what is the valency of phosphorus? 5. Am I right? If you can't keep on typing at the chat, you can use your mic, right? The valency of phosphorus is 5. Am I right?
Yes.
Yes. So let's see. 1, 2, 3, 4, 5. Correct, right? Correct.
Yes.
Now here there will be negative charge at oxygen, negative charge at oxygen, negative charge at oxygen. So see here, PO4 3 minus 1, 2, 3, 3 minus. Can you see? If you want, now if you find it difficult, sir, how does, do you have a question like this? Sir, how does that negative charge come? Do you have some sort of doubt like that? That is also chemistry part. Oh, are you clear with this? So we know that phosphorus, this is the formula and this is the charge. We know that. We are fine with that. Yes, you are fine with that. Are both all the students fine with that?
Yes, sir.
Yes. Okay, fine then. We can move on. Right. So can you understand phosphate group is the one that gives a negative charge because PO4 3 minus.
Yeah.
Yes. Fourth point, typically an additional polar molecule or charged molecule is also linked. What is the additional polar molecule or charged molecule? This choline. This is a small charged molecule. Slight positive charge is present for that. Not as phosphate. Now phosphate has 3 minus. This has a mild 1% like charge. Slightly polar molecule. Can you understand that? They haven't spoken much about choline here. Can you understand? Is it clear? Yes. Is it clear for all? One student has mentioned that she's clear with that. Clear, right? Very good, very good. Right. We are coming to the last 2 points. The two ends of phospholipid show different behavior. Two ends means fatty acid. Fatty acid contains what are the elements present in fatty acid? What are the elements?
Carbon, hydrogen, and oxygen.
Carbon, hydrogen, And oxygen, yeah, oxygen correct. But fatty acid, the tail, the chain,
Yeah, carbon and hydrogen only.
carbon and hydrogen, right? So fatty acid contains only carbon and hydrogen. If they say including the carboxylic terminal, what you said was correct because it contains oxygen. But here the chain, see here, they have mentioned it as hydrophobic tail. See here, hydrophobic tail. So only because it contains carbon and hydrogen, it is said to be hydrophobic, right? If you see phosphate, phosphate and choline, they are hydrophilic. Will you agree with that? Phosphate molecule is hydrophilic because they have a charge. They can react with water. Will you agree with that? Now, if you want to
Yes, sir.
clarify, I can explain it also because with the knowledge of the water knowledge, you have learned about water, right? Now, if this is phosphate, water is looking like this, right? We know that hydrogen tends to react with oxygen, forming what type of bond? What type of bond? Hydrogen bond, right? So it tends to react. So we can say that phosphate is hydrophilic or hydrophobic.
Hydrophilic.
Hydrophilic, very good, right? So we can you see this upper end shows what type of nature? Hydrophilic or phobic?
Hydrophilic.
Hydrophilic. This bottom part that shows hydrophobic or hydrophilic. The fatty acid.
Hydrophobic.
Hydrophobic, right? So can you see there is an opposite nature, hydrophilic, hydrophobic. Both the ends show different natures, right? And the last point, the hydrocarbon tails are hydrophobic, while phosphate group and its attachment head. Attachment head means what is it?
That choline.
Choline, exactly, right? Are hydrophilic. Is that clear? The phospholipid part. Just check me and let me know. Just check this complete part and let me know. Is that clear? This complete part. Yes, our one student has mentioned that clear power. Right, very good. Okay, then we are coming to the end, which is functions of lipids. Right, only there are only four. First one, food reserves and energy source. We know that lipids function as energy source. Example, triglycerides such as fats and oils. Normally, fats that are present in our skin, right under the skin, there are tissues called adipose tissues, right? Those tissues stores fat as energy. Can you understand? Now you will be learning this in unit number 5, which is will be when we will be learning about our human body. We'll be learning about those sections, right? Simply you should understand that lipids are acting as energy sources. Will you agree with that point?
Yes.
Yes. Second one, maintains the fluidity of plasma membrane. Yes, fluidity of plasma membrane contributed by phospholipid and cholesterol. Now, in this here I explained that plasma membrane looks like this, and it contains phospholipid molecules. And here there are cholesterol molecules also like this, right? This will be maintaining the fluidity of membrane. Fluidity means it helps in exchanging the materials like a fluid. Now, what does a fluid do? What does a fluid do? It helps in exchanging. Now, if you see blood, what does blood do? Blood helps in exchanging materials like a medium. So here also it helps in exchanging materials just because phospholipid molecules and cholesterol is present. Can you understand? Yes or no? One student has said it's clear. Is it clear? Yes. Okay. Third, acts as signaling molecules or hormones that travel through the body. Signaling molecules or hormones. Example, steroids. Steroids are functioning as hormones. Is steroid a type of lipid? Let's check.
Yes.
Yes, here steroid is. So I hope you have come across the word steroid. Now normally the ones who involved in gym activities, they are more familiar with the word steroids. Normally steroids they say they are taking injections. Normally they say steroids is illegal. In sports, athletics, steroids is illegal because it is a type of hormone that can induce your dopamine levels, right? Dopamine levels, the functioning body. There's an but if you see overall, it is illegal. Have you come across that? Normally in films they used to portray this. Have you come across that? No, something like that. No, which means no. I'm not trying to say that. Have you come across that situation in your real life? I'm saying that. Have you heard about this steroids? It's illegal to be used in Olympics.
Yes.
Yes. I guess during Susantika Jayasuriya's Susantika's the race, the track event which she participated also, she got the third place and the second place. The winner, the player who played, who won the second place, she also used some sort of steroid, so they disqualified her, and Susantika got the second place. There was a news spreading. Have you come across that news? No. Yes. You have come across, right? So there was a scenario like this also. So it is illegal to be used. Right. Last one, found as components of animal cell membrane. Correct, right? Cholesterol. It is present in animal cell membrane as well. Normally, right? Cholesterols are present in animal cells also. But now you might ask, sir, you said cholesterol is bad, right? But this is present in the human body cell membrane itself, right? Now, if you see cell is present in the cell membrane, if it present, it is okay. But if you see blood and blood vessels, they are very sensitive. For blood and blood vessels to be transmitted, if cholesterol comes and blocks like this, anything that blocks the functioning, it is harmful. Now, if you see here plasma membrane, cholesterol is remaining as it does not affect anything and it just remains. It is helping in its function. But here, is it helping in its function? It is blocking and it is affecting the function. Can you see? Can you see?
Yeah.
Yes. Okay. Just go, and that's the end, right? Just go through these functions, and if it's completely clear, then that's it for the day. Hopefully, next class we'll start protein. Just check and let me know. Clear. One student has said that it's clear for her. No hurry, just check and let me know. Right? Mm. Is it clear? All 4 points? Mm. Is it clear for all?
Yes, sir, clear.
Yes. Okay then. Okay then. Uh, that's it for the day. Thank you for joining live, right? Because some students, normally watch the recording, so I'm not speaking anything bad about them. They have a busy schedule and they are watching the recordings, but joining it live and supporting me, thank you for it. And hopefully, we'll be starting proteins next week. So before coming next week, completely revise this section, right? So if you allocate 30 minutes per day, it's enough. You can complete this revision within 2 to 3 days. That's it, right? Okay then. Hopefully I'll give an essay tomorrow in the group, right? So you can try it, and the online students can send it as a photograph to me, or you can request me for the scheme. I'll provide it. Physical students, you can write the essay and you can give it when we meet it physically next Friday, right? Okay then. Thank you for joining. That's it for the day.
So how to get your group link?
Group link. I'll wait. I'll share it in this group chat. Wait.